Good afternoon, everyone. Today I will be talking about motor unit summation and tendinization of muscles. So what is a motor unit exactly? All muscle fibers are innervated by a single nerve fiber. Each motor neuron that leaves the spinal cord innervates multiple muscle fibers. Together, they make up the motor unit. Usually, small muscles that react rapidly and whose control must be exact, such as finger movements, have more nerve fibers for fewer muscle fibers, while large muscles that do not require fine motor control, such as your biceps femoris, may have several hundred muscle fibers in a motor unit. The muscle fibers in each motor unit are not all bunched together in the muscle, but overlap other motor units in little bundles of 3 to 15 fibers. This interdigitation allows the separate motor units to contract in support of one another rather than separately. Next, we're going to talk about muscle fiber summation. So what is summation? Summation means adding together of individual twitch contractions to increase the intensity of the overall muscle contraction. Just like adding in algebra. Adding twitch contractions. This happens in two ways, by increasing the number of motor units contracting simultaneously or by increasing the frequency of contraction. Increasing the frequency would be tendinization. So how does summation occur exactly? When the CNS sends a weak signal to contract a muscle, the smaller motor units of the muscle may be stimulated in preference to the larger ones. As the strength of the signal increases, larger and larger motor units begin to be excited as well. The larger motor units often have as many as 50 times the contractile force of the smallest units. This is the size principle. So why is the size principle important? Because it allows the gradations of the muscle force during weak contraction to occur in small steps, while the steps become progressively greater when large amounts of force are needed. What causes the size principle is that the smaller motor units are driven by small motor nerve fibers, and the small motor neurons in the spinal cord are more excitable than the larger ones, so they are excited first. Another important feature of multiple fiber summation is that the different motor units are driven by the spinal cord, so contraction alternates among motor units one after the other. So it's not consistently using one motor unit. That would tire it out. This provides smooth contraction even at low frequencies of nerve signals. Next, we will cover frequency summation and tendinization. If you look at this figure, it shows the basic principles of these two concepts. To the left are shown individual twitch contractions occurring one after the other at low frequency. As the frequency increases, there comes a point where each new contraction occurs before the preceding one is over, as you can see here. The result of this is the second contraction is added partially to the first, so the total strength of the contraction rises progressively with increasing frequency. When the frequency reaches a critical level, the successive contractions eventually become so rapid that they fuse together and the whole muscle contraction appears to be completely smooth and continuous. This is tendinization. At a slightly higher frequency, the strength of contraction reaches its max. Any additional increase in frequency beyond this point will have no further effect in increasing the contractile force. Now we're going to talk about the neuromuscular junction. So what is the neuromuscular junction exactly? The nerve fiber forms a complex of branching nerve terminals that invaginate into the surface of the muscle fiber, but lie outside the muscle fiber plasma membrane. This entire structure is called the motor end plate. The end plate is covered by one or more Schwann cells that insulate it. As you can see here, the Schwann cells are making up the myelin sheath. In the axon terminal are many mitochondria that supply ATP that is used to make an excitatory neurotransmitter, acetylcholine. The acetylcholine then excites the muscle fiber membrane. So let's talk about acetylcholine for a minute. How is it released exactly? When a nerve impulse reaches the neuromuscular junction, about 125 vesicles are released into the synaptic space. As you can see here, there are linear dense bars inside. These are the dense bars. On each side are protein particles that penetrate the neural membrane, the voltage-gated channels. These are the voltage-gated channels for calcium. When an action potential spreads over the terminal, these channels open and allow calcium ions to diffuse from the synaptic space to the interior of the nerve terminal. The calcium ions draw acetylcholine vesicles to the neural membrane adjacent to these dense bars. So they're going to pass around the dense bars and diffuse into the space. The vesicles then fuse with the neural membrane and empty their acetylcholine in the synaptic space by exocytosis. But how is acetylcholine destroyed? Once it is released, it continues to activate the acetylcholine receptors as long as it is present in the synaptic space. It can be removed in two ways. One way is through acetylcholinesterase, which is an enzyme that will destroy it and make it no longer available for use, or small amounts of acetylcholine will diffuse out of the synaptic space and they will no longer be available for muscle fiber